Welcome to the Deep Dive. Today we're looking at, well, what feels like a really major medical breakthrough. It certainly got people talking, especially those tracking genetic therapies.
Yeah, it feels different, doesn't it? More than just a small step. It feels like maybe the start of a revolution in personalized medicine.
Exactly. And at the heart of it is this tool we've discussed before, CRISPR.
Right, CRISPR. Amazing technology, won the Nobel just over 10 years back. It essentially lets scientists, cut and paste bits of DNA like a word processor for genes.
And the dream has always been, can we use this to actually cure genetic diseases?
That's been the promise for years. And now, well, it looks like that future might be arriving.
So today that's our mission. We want to really understand what this news means, its implications, all through the story of one specific baby, known as KJ.
A very personal story and quite incredible.
Let's get into it then. KJ's journey. He was born late 2024, parents Kyle and Nicole Muldoon, at the Children's Hospital of Philadelphia.
And right away there were problems, serious ones.
Yeah, the doctors knew something was wrong. He wasn't eating, he was vomiting constantly, and then these terrible epileptic seizures started.
Just imagine how terrifying that must have been for his parents.
Aye.
A newborn baby.
Absolutely. So they did genetic testing.
And the results came back with, well, a devastating diagnosis. KJ had inherited 2 faulty gene copies, one from each parent.
Leading to this condition, carbamoyl phosphate synthetase 1 deficiency, CPS1 deficiency.
Which is, first, incredibly rare, and second, until this point, completely incurable.
And when you say rare, you mean really rare.
Extremely. We're talking maybe 1 in 1.3 million babies, something like that.
Wow.
And the prognosis is grim. About half don't survive the first week.
That's shocking.
It is. And those who do survive, they often face really severe developmental issues, mental delays. Most end up needing a liver transplant.
Which itself means a lifetime of immunosuppressant drugs, constant checkups.
Right. It's just a relentless condition, unimaginable stress for the family.
So what's actually going wrong inside the body? Why is it so severe?
Okay, so it boils down to how we process proteins. Our bodies need protein, obviously, but breaking it down creates a waste product, ammonia, which is toxic.
Right.
Normally, your liver has an enzyme, this CPS1 enzyme, that takes the ammonia and converts it into urea, something harmless that you just, pee out.
Okay, simple enough.
But KJ's mutations meant his body couldn't make that enzyme. No CPS1.
So the ammonia just builds up.
Exactly. It accumulates in the blood, and it's particularly damaging to the brain. That's what causes the vomiting, the not eating, the seizures. It's poisoning him, essentially.
So with that diagnosis, what could they even do? What were the options on the table? Very few, I'm guessing.
Very few. And none of them were cures. It was all about management. First, a super strict diet, minimal protein intake, just trying to keep those ammonia levels down.
Must be incredibly hard with a baby.
Extremely difficult. Plus, he needed injections of a drug, glycerol phenylbutyrate, which helps the body get rid of some ammonia.
But even with all that,
there were no guarantees. Survival was still very much in question. It was a truly desperate situation for his parents.
okay, so things look incredibly bleak. But then the story shifts.
It really does. This is where hope starts to enter the picture. KJ's doctor, Rebecca Ahrens-Nicholas, she reached out to a researcher, Kiran Musunuru.
And Musunuru had an idea, a radical one. He described it himself as risky but possible.
Yes. And what he proposed was using CRISPR gene editing to actually go into KJ's DNA and fix the errors.
Fix the typos in his genetic code, so to speak.
Exactly. Our DNA is like the instruction manual for building and running our bodies. Diseases like KJ's happen because there's a mistake, a mutation in those instructions.
So CRISPR could potentially correct that specific mistake.
That's the power of it, yes. Not just managing the symptoms, like the ammonia buildup, but fixing the root cause the faulty gene itself, correcting the specific letters in his DNA code that were causing the problem.
But this wasn't something routine. This was experimental.
Highly experimental, and they were running out of time.
Right, the clock was ticking. They only had months, maybe, before irreversible brain damage could occur.
So they had to act incredibly fast. Unprecedented speed, really.
Which brings us to how they actually did it. This wasn't just an off-the-shelf treatment.
Not at all. It was completely personalized for KJ. Developed in 3 main phases. Phase 1 was all about lab work, testing, getting the precision right.
They used KJ's own cells.
Yes, they took samples of his cells to test different versions of the CRISPR treatment. And the specific CRISPR technique they used was quite advanced. Not just cutting the DNA, but a variant called base editing.
Base editing.
Yeah, it allows for changing just a single DNA letter to another one, which was exactly what was needed for KJ's specific mutation. Super precise.
Okay, precision tool identified. But how do you get that tool into the right cells in the liver? That seems like a huge challenge.
It is. You need a delivery system. And they use something you might have heard about from the COVID vaccines, lipid nanoparticles.
All those tiny fat bubbles.
Exactly. They act like little delivery vehicles, carrying the CRISPR machinery safely inside the target liver cells.
Clever.
And during this testing, they found something interesting. The mutation KJ got from his father was actually a bit easier to correct than the one from his mother.
Does that matter? Don't you need both copies fixed?
Often, no. For many genetic diseases, having just one working copy of the gene is enough to restore function and stop the symptoms. So they decided to focus their efforts on correcting the father's mutation.
Makes sense. Target the easier fix first.
Right. And this whole phase, the lab work, the testing, the refinement, it involved this incredible global effort.
You mean multiple teams working together?
Yes, research groups from all over the world basically dropped what they were doing to pitch in, sharing data, results, ideas. They condensed what normally takes many months, maybe even years of research into just 1.5 months.
That's astounding. For one baby.
It shows what's possible when people collaborate intensely on a critical goal. The urgency just pushed everyone forward.
So lab phase done incredibly quickly. What's phase 2?
Phase 2, animal testing. You can't go straight from a petri dish to a human baby. So first, they created what's called a humanized mouse.
A mouse with KJ-specific gene mutations.
Exactly. They engineered mice to have the same CPS1 mutations, essentially replicating the disease in the lab. They gave these mice the treatment.
Did it work?
It did. A single dose corrected the gene in up to 25% of the liver cells. And crucially, there were no significant side effects observed.
25%, is that enough?
It seemed promising, yes. Often you don't need 100% correction. So the next step was testing in larger animals closer to humans.
Primates.
Yes, they tested the treatment on 9 macaques. Again, the key things were, is it safe? Is it non-toxic?
Yeah.
And does it actually reach the liver effectively? The answer to all those questions was yes.
Okay, so lab tests look good, animal tests look good. The path seems clear for the final, most critical step.
Phase 3. Treating KJ himself. By this point, about 6 months had passed since the research sprint began. KJ was 7 months old.
Must have been an incredibly tense moment.
Absolutely. They started with a very small, cautious first dose, just to see how his body would react. Safety first.
And the reaction.
It was almost immediate. And positive, dramatically so. His ammonia levels started to drop.
Wow.
And he started tolerating more protein in his food without the vomiting, without the negative effects.
That's incredible, just from the first dose.
Yes. Then they gave him a second injection. After the second shot, his ammonia levels fully normalized, back to healthy baby levels. He could eat normally.
Amazing.
They gave him a third and final dose, just to be sure. And then, after 8 long months living in the hospital, KJ went home.
He finally got to leave the hospital.
He did. He's 10 months old now and apparently doing remarkably well recovering.
What an incredible outcome.
Okay.
So let's step back. Why is this specific case being hailed as such a landmark? We've heard about CRISPR successes before.
Right. This feels different. It's what some are calling a bombazo, a real bombshell, because it's the first time CRISPR gene editing has been used after birth to fix a specific mutation causing an incurable metabolic disease in a human.
Curing it at the source.
Exactly. Not just managing symptoms, but correcting the fundamental genetic error. This is like the ultimate example of personalized medicine, a treatment designed and delivered for one specific person's unique genetic makeup.
Okay, but some people listening might be thinking, didn't we hear about CRISPR being used on babies years ago?
Well,
That Chinese scientist.
Yes. You're probably thinking of He Jiankui in 2018. That was hugely controversial, and for good reason.
Right.
He used CRISPR on embryos before the babies were born, trying to make them resistant to HIV. That editing affected all their cells, including their reproductive cells, meaning it could be passed down generations. And it was done secretly, unethically.
So very different from KJ's case.
Completely different. KJ's treatment was after birth. It targeted only his liver cells, the source of the problem. And the goal was clearly therapeutic to cure a life-threatening disease he already had, not enhancement, not germline editing.
And we have seen other CRISPR milestones, haven't we? Treating genetic blindness.
Yes, Leber congenital amaurosis back in 2019. That was a big step.
Fine.
And more recently, the approval of Casgevy in 2023.
For sickle cell anemia and beta thalassemia.
Right. Those are treatments that modify stem cells outside the body and then put them back in. Hugely important, but still perhaps managing the disease process. KJ's case feels like a step further, a direct in vivo correction, a cure from the origin point.
Fixing the gene inside the body.
Precisely.
So looking ahead now, this is obviously a massive breakthrough. But what are the remaining questions? It can't be all settled, right?
No, absolutely not. There are definitely unknowns. A big one is durability. Will this fix last? Will the edited genes keep working properly in KJ's liver cells as he grows older?
Could the effect wear off?
It's possible. We don't know. Will he need more treatments later in life? Or could he still, despite this success, eventually need a liver transplant? These are questions only time and careful monitoring of KJ will answer.
And then there's the elephant in the room. Cost and scalability.
Right. This was, as you said, a brutal effort. Multiple world-class research teams, immense resources, all focused on one child.
The cost must have been astronomical, even if we don't know the exact figure.
Undoubtedly. And there are over 7,000 known rare diseases. Replicating this kind of intensive, bespoke effort for every single one, or even many of them, it's just not feasible right now. The economics are a huge hurdle.
But is there reason for optimism on that front?
I think so, yes. Look at the cost of sequencing a human genome.
Okay.
Back in 2003, the Human Genome Project finished cost something like $2.7 billion.
Billion with a B.
Right. Today, you can get a whole genome sequenced for under $1,000, maybe even less soon.
That's an incredible drop.
It is. And that kind of technological advancement, making things faster, cheaper, more efficient, it suggests a similar path might be possible for gene editing therapies. The tools will get better, the process is more streamlined, the costs will likely come down.
So diagnosing these rare conditions gets easier, and maybe designing the personalized fixes becomes more manageable too.
That's the hope. It won't happen overnight, but the trajectory seems positive. This case, KJ's case, is truly a giant step for personalized medicine. And, it's also just an amazing story of scientific collaboration. What can be achieved when people really pull together?
Absolutely inspiring. It really shows the potential.
It does.
So KJ's story really is a landmark. A glimpse maybe into a future where genetic diseases we once thought were incurable might actually be fixed, using treatments tailored to the individual.
As we wrap up, thinking about what comes next, what do you see as maybe the biggest immediate challenge, or perhaps the next disease target this kind of approach might unlock? Well, the challenge is definitely scaling, as we discussed, making it accessible. As for next targets, there are many other single-gene metabolic disorders affecting the liver that could theoretically be approached this way. But honestly, each one presents its own unique challenges. It's still early days.
Early days, but incredibly promising ones. It leaves you wondering, doesn't it? How close are we really to tackling other major genetic diseases? And what are your own thoughts on the power and maybe the risks of these increasingly precise CRISPR tools? Something to think about.